Hello everyone, welcome to the lecture on Chandipura virus, an emerging tropical pathogen. First, let us look into the historical perspective. Chandipura virus closely resembles vesicular stomatitis virus. Vesicular stomatitis has been known for more than a century as a disease that causes severe mouth lesions in cattle, with ruptured vesicles in the gum or tongue and reddish ulcerations. Chandipura virus is readily distinguished by its ability to infect humans. It was first discovered in 1966 by Butt and Rodericks while investigating patients suffering from fever in Chandipura village in northern Maharashtra near Nagpur district. The virus was named after its place of isolation as Chandipura virus. Subsequently, in 1980, Chandipura virus was isolated from the serum of an encephalopathy patient. This further confirmed its existence and its ability to propagate in the human population. It was only in 2003 that the first evidence for its association in human epidemic was obtained during acute encephalitis outbreaks. It has garnered global attention as an emerging neurotropic pathogen, inflicting high mortality in children within 24 hours of commencement of symptoms. Epidemiology. Till date, the presence of this virus is recorded from the Indian subcontinent, India, Bhutan, and Nepal, Sri Lanka, and Africa, Nigeria, and Senegal. One outbreak took place in the state of Andhra Pradesh and affected 329 children, resulting in 183 deaths. Simultaneously, another encephalitis outbreak was reported in the state of Maharashtra, resulting in about 400 encephalitis cases with 115 deaths. Subsequently, another outbreak of encephalitis associated with Chandipura virus infection was reported in 2004 in the eastern state of Gujarat with a fatality rate of more than 75%. These recent outbreaks indicate the emergence of Chandipura virus as a deadly human pathogen in the Indian subcontinent. The most recent outbreak reported was from the state of Gujarat in 2019, where a 5-year-old girl succumbed to the infection caused by the Chandipura virus. The prevalence of anti-Chandipura virus antibodies in animal sera indicates that domestic animals might serve as a natural reservoir for this pathogen. Indeed, the presence of neutralizing antibodies to Chandipura virus was confirmed in 30.6% of pig, 14.3% of cattle, 17.9% of buffalo, 9.3% of goat, and 7.7% of sheep serum samples collected from the epidemic affected areas. The female sandflies, phlebotomine sandfly, ticks, and mosquitoes are proposed to be the Chandipura virus carriers, wherein this arthropod virus resides in the salivary gland of these insects and is transmitted to mammalian hosts through bites. The Chandipura virus is transmitted through sandfly, especially where cattle are kept in unclean cattle sheds. Human beings get the virus when the flies bite at night. Structure of the Chandipura virus. Chandipura virus belongs to the genus Vesiculovirus, placed in the order of Mononega of Rhabdoviridae family. Chandipura virus is an enveloped RNA virus with an approximate genome length of 11 kb. It is 150 to 165 nanometer long and 50 to 60 nanometer wide, showing distinct surface projections of 9 to 11 nanometer in size and a stain-filled canal at the base of the virus particle. These are the transmission electron micrographs of Chandipura virus isolates from tissue culture. These are two negative stain virus particles showing the stain-filled canals and basal attachments. This is a negatively stained Chandipura virus particle showing typical vesiculovirus morphology, including the internal ribonucleoprotein coil. Chandipura virus is surrounded by lipoprotein envelope, which encloses a helical ribonucleoparticle with a non-segmented single-stranded RNA enwrapped by nucleocapsid protein N. Components of the viral RNA-dependent RNA polymerase, large protein L, and phosphoprotein P are also packaged and remain associated with the core nucleocapsid particle. Glycoprotein G protrudes externally from the outer membrane while matrix protein M lies in the inner bilayer of the membrane. Genome organization of the Chandipura virus. The genome of Chandipura virus is a single-stranded negative sense RNA. The genomic RNA comprises of 49 nucleotide leader gene L followed by 5 transcriptional units coding for viral polypeptides separated by intragenic spaces and a short 46 nucleotide non-transcribed region called as a trailer sequence. The genome is arranged as a short leader sequence at the 3' end L, the gene N which codes for the nucleocapsid protein, the gene P which codes for the phosphoprotein, gene M which codes for the matrix protein, gene G which codes for the glycoprotein G, and gene L which codes for the large protein L, the trailer sequence T at the 5' region. The nucleocapsid protein N is a 422 amino acids long polypeptide of 49 kilodalton coded by the N gene. It is the most abundant viral protein in infected cells. It binds with the nascent leader RNA, thereby encapsulating RNA genome in a manner just like histone-mediated enwrapping of a DNA molecule into a nucleosome structure. This encapsulated genome is used by viral polymerase as a template during transcription and replication. The N protein protects the viral genome from host ribonucleases and plays regulatory rules in transition from transcription to replication in the virion life cycle. Hence, it is designated as the transcription replication switch. Phosphoprotein P is a 294 amino acid long polypeptide of 29 kilodalton coded by the P gene. P together with the L protein constitutes the viral RNA-dependent RNA polymerase, where L is a catalytic subunit and activator of viral transcription and P is the cofactor. Matrix protein M. M protein is a 229 amino acid long polypeptide of 26.3 kilodalton. It lies in the inner bilayer of the membrane and acts as a bridge between the encapsulated genomic RNA called the nucleocapsid and the outer membrane envelope. M protein also interacts with and recruits mature nucleocapsid proteins to the membrane during viral assembly and budding. Matrix protein of the vesicular stomatitis virus functions in viral assembly, was shown to stop post-transcription by RNA polymerase 1 and 2. This M protein mediated inhibition of host gene expression and it constitutes in the suppression of cellular interferon response. Glycoprotein G is a 59.5 kilodalton protein that protrudes externally from the outer membrane and acts as a major antigenic determinant, and therefore it elicits an antibody response. It is a sole spike protein of Chandipura virus that contributes in the absorption, assembly, and final budding of the newly formed viral particle. Vesiculovirus glycoprotein G exists as a trimer which reversibly adopts three distinct conformational states. The native state present on the virus surface which is stable above pH 7, the activated state that fuses with the target membrane, a fusion inactive post-fusion state that is stable under low pH condition. The glycoprotein G of Chandipura virus is an ideal candidate for vaccine development. The large protein L is encodes for the largest viral protein of 2092 amino acids of 238.5 kilodalton. Together with P, it constitutes the viral transcriptase. It persists the catalytic activity of RNA polymerization, capping, and polyadenylation. L shows the protein kinase activity and also associated with cellular translation elongation factors. Ribonucleoprotein particle is composed of core nucleocapsid and is associated with viral RNA-dependent RNA polymerase subunits. The ribonucleoprotein particle is infectious by itself because it contains all the enzymatic activity necessary for the synthesis of RNA and could produce progeny viral particles when introduced artificially within the cell. Chandipura virus gene expression illustrates two distinct well-conserved characteristics, namely sequential and polar transcription. Vesiculovirus mRNAs are synthesized in an obligatory sequential manner after polymerase entry at a single site at the 3' end of the genome, that is, the beginning of the leader RNA. Measurement of molar ratios of different vesicular virus mRNAs indicated that their abundance decreases with increasing distance from the polymerase entry site in the order nucleocapsid protein is synthesized more than the phosphoprotein, which is expressed more than the matrix protein M, which is expressed more than the glycoprotein G, and the glycoprotein G is expressed more than the large protein L. To explain this polar and sequential mRNA synthesis, a stop-start model has been postulated for vesiculovirus transcription. In this model, subsequent entry into the nucleocapsid RNA template, the viral RNA-dependent RNA polymerase sequentially transcribes the genome with progressive attenuation at each gene boundary, which results in decreasing amount of transcripts for genes that are distant from the entry site. Each termination event may cause the polymerase to fall off the template or may allow for reinitiation at the downstream promoter. Therefore, transcription of the downstream gene depends on the termination of the upstream gene transcription and reinitiation process. Genomic RNA enwrapped in the nucleoprotein N into the nucleocapsid structure and is not the naked viral RNA that is recognized by the polymerase as a template for RNA synthesis. The viral RNA-dependent RNA polymerase, composed of L and P proteins, possesses catalytic activities for RNA polymerization, capping, polyadenylation. The protein P plays an essential role in viral transcription. The P protein, through its C-terminal domain, interacts with both the nucleocapsid RNA template and the L protein, thus assembling transcriptase complex on the genomic template. The P protein utilizes its N-terminal acidic domain to interact with and activate polymerase function during transcription, similar to the activation mediated by classical eukaryotic transcription activators. The Chandipura virus P protein exists in the viral infected cells in two distinct states. The unphosphorylated form, that is designated as P0, while the phosphorylated counterpart is referred to as P1. Phosphorylation of P at a distinct site is required for P to act as a transcriptional activator. One unique feature of the vesicular virus gene expression is the utilization of a single L protein for mRNA synthesis, 5' end modification and 3' end polyadenylation of the mRNA. With the exception of leader RNA, all viral transcripts possess a 5' end cap structure similar to that observed in eukaryotic cellular mRNAs. Although certain host factors have been postulated to participate in capping reaction, primary enzymatic activity that mediates mRNA capping resides within the L protein. Viral transcriptase is also involved in the polyadenylation of viral mRNA. During transcription, the viral polymerase obeys termination signals present at the gene boundaries to produce discrete viral mRNAs, whereas the same polymerase anti-terminates at gene boundaries during replication phase to produce a positive sense polycistronic complement of the entire genome. Subsequent rounds of replication that utilize the positive sense replication intermediate as a template produce many more copies of the progeny negative sense genomic RNA, which after packaging buds out as a mature virion. Now let us see what is the positive sense RNA and negative sense RNA. Positive sense RNA is a viral RNA that is similar to mRNA and thus can be immediately translated by the host cell. However, negative sense viral RNA is complementary to mRNA and thus must be converted to positive sense RNA by an RNA polymerase before translation. The switch in polymerase function that allows it to ignore termination signals during replication is known as the transcription replication switch. Although the same L protein is involved in RNA polymerization during both transcription and replication, the phosphorylation state of the regulatory subunit P is proposed to be different in the two processes. It is postulated that the formation of L-N-P0 complex enables L protein to recognize promoter elements for replication and suppress transcription termination signal present at the gene boundaries during the viral replication. The form of P protein, P1, is engaged in transcription appears to be distinct from the form P0 that has been implicated in replication. Viral replication intermediates or replication products always remain encapsulated by N protein within infected cells and enjoy protection from cellular RNases. Consistently, vesicular virus replication critically relies upon de novo synthesis of the nucleocapsid protein N. However, N protein is prone to aggregation and this aggregated form is unable to appropriately encapsulate the viral RNA. Phosphoprotein P, by the virtue of its capsid-specific chaperone-like activity, prevents self-aggregation of the N protein, and maintains it in an encapsulation competent state. Now, let us look into the life cycle of Chandipura virus. Chandipura virus glycoprotein G binds with the host receptor and gets internalized through endocytic vesicles. With the lowering of pH within the vesicles, the ribonucleoprotein gets released into the host cytoplasm. The primary transcription of viral mRNAs begins. The newly synthesized glycoprotein G migrates into the endoplasmic reticulum, where it is packaged and secreted out in the host plasma membrane by the Golgi complex, while the translated nucleoprotein N, phosphoprotein P, and the large protein L take part in genome replication. And secondary transcription, the P protein acts like a switch that decides between replication and transcription. The translated M protein, like the G protein, also migrates to the plasma membrane but to the inner side. The newly synthesized viral RNA is enwrapped in the N protein along with L and P and gets ready to be released out of the cell. The glycoprotein G gets attached to the lipid envelope and completes the formation of a mature virion. Symptoms of Chandipura virus infection. Children below 15 years of age are more vulnerable than adults who show symptoms similar to any encephalitis case, such as high-grade fever, vomiting, altered sensorium, unconsciousness, coma, and convulsions. Altered sensorium is a medical condition characterized by inability to think clearly or concentrate. Convulsions are rapid involuntary muscle contractions that cause uncontrollable shaking and limb movement. Diagnosis of Chandipura virus infections. Chandipura virus infections can be diagnosed using immunofluorescent antibody technique. This is a microscopic method that can detect and visualize viral proteins expressed in cells via antigen-antibody reactions. Sandfly and mosquito cell lines can be employed for early detection of Chandipura virus, as the antigen could be detected within 2 hours of inoculation using immunofluorescent antibody technique. Characteristic cytopathic effects produced by Chandipura virus in vertebrate cell lines also help in detection of the virus. Cytopathic effects are the changes in cell morphology caused by virus infection. Common examples of cytopathic effects are rounding of infected cells, fusion with adjacent cells to form syncytia, that is polykaryocytes, and the appearance of nuclear or cytoplasmic inclusion bodies. Reverse transcriptase PCR and real-time PCR have also been standardized for routine diagnosis and are the most reliable methods for detecting the Chandipura virus. On the basis of sample investigation, Chandipura cases can be categorized as a possible case, a patient meeting clinical criteria. A probable case is defined as a patient with a sudden onset of high-grade fever followed by central nervous system involvement, altered senses, convulsions, or a comatose state, singly or in combination, and negative test results for malaria and other common causes of illness. Confirmed case. A confirmed case is defined as a patient with one or more of the following laboratory results: presence of viral RNA as detected by RT-PCR or real-time PCR, IgM antibodies against the Chandipura virus in a clinical sample, seroconversion in a convalescent sample. Seroconversion refers to the time between the exposure to a virus and when antibodies show in blood. Convalescent sample is a blood sample that is obtained from an individual who has recovered from the Chandipura disease and contains antibodies against the Chandipura virus, and also by viral isolation and detection of the cytopathic effects in cell lines. Now, let us look into the vaccination aspects of Chandipura virus. A recombinant vaccine using the complete glycoprotein gene, the G gene of Chandipura virus, has shown good protective response in mice. A beta-propiolactone inactivated tissue culture based vaccine has also shown excellent protection in mice upon booster vaccination. siRNAs which silence the phosphoprotein gene P and the matrix protein gene M gene of the virus has also shown promising results both in vitro and in vivo. However, clinical trials have not been conducted for any of these. As a result, there are no commercial vaccines against the Chandipura virus. Now, let us look into the treatment options available to curb the Chandipura virus. There is no specific treatment available to date. Symptomatic treatment involves use of mannitol to reduce brain edema caused by the virus infection, containment of disease transmitting vectors, maintaining good nutrition, health, hygiene, and awareness in rural areas will help in curbing the menace of the Chandipura virus. Vector control as a preventive measure. Spraying of insecticides could bring down the population of the sandflies drastically. However, control of vectors in endemic areas is difficult as they breed in damp places inside crevices of stone used for construction where insecticide spraying is generally not possible. Cow dung smearing of the floor and walls of houses is another practice of the inhabitants of endemic areas, and they accumulate dried cow dung sheets inside the houses as fodder. It has been reported that cow dung serves as a feed for sandfly larvae and thus helps in the spread of the Chandipura virus infections. Now, a brief summary. The virus was first isolated and identified in 1965 at Chandipura village in Nagpur, hence the name Chandipura virus. It is transmitted to humans by sandflies that breed in small dark crevices and cracks of houses. 58% is the fatality rate associated with Chandipura virus infection. The virus mainly affects children. The virus was not considered to have an epidemic potential until an outbreak of acute encephalitis happened among children in Andhra Pradesh in the year 2003. The disease progresses from influenza-like symptoms to coma, resulting in death in extreme cases. Viral encephalitis is a public health concern worldwide. No specific treatments and vaccines are available to treat the Chandipura virus infection. Hence, prevention is the best way to control the viral infections. These are a few references and journal articles on the Chandipura virus for further reading. Now, let us discuss some multiple choice questions on the Chandipura virus. The Chandipura virus belongs to which family of viruses? A. Flaviviridae, B. Togaviridae, C. Bunyaviridae, D. Rhabdoviridae. The correct answer is D. Rhabdoviridae. The Chandipura virus is a bullet-shaped virus which belongs to the family Rhabdoviridae. Where was the Chandipura virus first identified? A. Africa, B. South America, C. India, D. Southeast Asia. The correct answer is C. India. What is the role of the Chandipura virus glycoprotein in viral life cycle? A. Viral entry, B. Viral assembly, C. Viral release, D. Viral transcription. The correct answer is A. Viral entry. Which structural protein of Chandipura virus forms the helical capsid surrounding the viral RNA? A. Nucleoprotein, B. Glycoprotein, C. Matrix protein, D. Polymerase. The correct answer is A. Nucleoprotein. What is the primary route of Chandipura virus dissemination within the host? A. Blood circulation, B. Lymphatic system, C. Nervous system, D. Respiratory system. The correct answer is C. Nervous system. What is the primary outcome of Chandipura virus infection in humans? A. Encephalitis, B. Respiratory distress, C. Gastroenteritis, or D. Hemorrhagic fever. The correct answer is A. Encephalitis. What is the main enzyme involved in Chandipura virus replication? A. DNA polymerase, B. RNA dependent RNA polymerase, C. Reverse transcriptase, or D. Helicase. The correct answer is B. RNA dependent RNA polymerase. Thank you for your kind attention.